Yes, hello, good afternoon, good morning, welcome to this Genomatix webinar. My name is Christian Zinser and I will give you an introduction to the Genomatix Pathway System in the next hour. A few technical things before we start. You can ask questions by typing them into the questions box which you find in your GoToWebinar panel. Everybody is muted so that we can have good sound quality. Should any problems arise during the webinar, you can also just enter something into the chat box. For example, if the sound is not good enough, etc., and I will try to take care of that. Okay, so yes, I will answer questions at the end of the webinar if there is time, and any questions I can't answer, then I will try to answer per email. All right, so an introduction to the Genomatix Pathway System. I'll like to start with a few introductory slides and then actually go into a demonstration of, well, a lot of different features of the pathway system. So this Genomatix Pathway System, what does it do? It takes a list of input genes with optional numerical values, for example, expression values or maybe also expression changes, and it performs a gene enrichment analysis, for example, for pathways, canonical pathways, literature mining based pathways, gene ontology, and a number of other things. You can generate networks in the system using the different terms and the different biological categories which have been analyzed by the system, and also with a number of other filter settings, and you can combine filters. We have interaction information based on co-citation in PubMed. And for the canonical pathways, certainly also directly from the canonical pathways, both automatically curated interaction information and also expert curated interaction information. And you can also change your networks, manipulate them in a number of different ways. For example, change the layout of the networks or extend the networks. And also we have a host of transcription factor and transcription factor binding site information in the system. For example, transcription factor gene relationships, which are based on ENCODE chip data, and this is also shown and visualized in the networks, and you can access the corresponding information. Okay, so here is an overview of well, what we will cover. We'll start with the basics. So what are the data you input into the system and what are the parameters that you can set? Then I will show you a quick overview of the result page in the Genomatix Pathway System here, short GePS, and the user interface. I'll show you the gene ranker list, which is the list of the biological categories and the overrepresented terms within these categories based on your input genes. I'll show you a little bit on how you can filter this list to search for specific information, and how you can access also the hierarchical tree in the gene ontology, as we have gene ontology also in, as one of the categories in there. Then I'll proceed to the networks. Canonical pathways on the one hand and co-citations networks on the other hand, so what's the difference between the two. Then how you can access detailed information on interactions, detailed information on genes. I will show you a short example how you can get information on the transcription factor binding sites in a promoter of a gene. Then how to read the lines and arrowheads that make up the network connections, and also an overview of the transcription factors network, which you can show as a table. And the last part will be how you can manipulate the networks, change basic parameters, how the network is generated, how you can extend the network with a number of different nodes. So start with the microRNAs, then I'll show you manual extension with genes. I'll show you how you can use the literature, access the literature to add information to the network in the form of user-defined connections, and how you can extend the network with transcription factors, and lastly, how you can connect or add connecting gene nodes to a network so that you get a connection, short as possible connection via co-citation between any two genes. Okay, so let's go to the program itself. You find the Genomatix Pathway System in the Genomatix software suite here under Literature and Pathways, the first entry. And you have a number of different ways how you can run the pathway system. I will concentrate on the first option here, characterization of gene sets. If the other options are, you can also just enter one gene and let the system give you the approximately 50 most heavily co-cited genes for your gene or for a term. You can get the canonical pathways for a gene. You can just browse human pathways without any gene input, and you can also manually build a network from scratch. Okay, let's see what the characterization of gene sets lets you do. Here we are at the input. How do we get the data into the system, and what are the parameters that we can choose? So the data can either be input as a list of keywords, which you would enter in here. You need to select what type of keywords you have. You can use gene IDs from Entry or from Ensemble. You can even mix them. You can use gene symbols, transcript accession numbers, or probe set IDs from Affymetrix. Alternatively, you can also upload a text file, and that's what I will do. I just show you the content of the text file. It's actually here. So this is just a tab separated text file. We have the identifier here, in this case Ensemble, gene identifiers for genes in the first column, and we have log 2-fold changes from an experiment, which I will explain when we started the analysis. In the second column, we also have some number of identifiers for microRNAs in the system, which are down here. These are actually NCBI gene IDs for these microRNAs. So you can mix types of identifiers. You just need to be in the same category, so it's a gene ID in any case. Okay, let's just switch over to the pathway system once more. So I'm going to select this file. Here from this directory and just load it into the system. Okay, so these are our data. What parameters do we have? Important parameter here is what kind of annotation types we have available. By default, everything is activated here for human. If you work with other species, then there might be fewer of them. Human is the most heavily annotated organism in our system. If you have another vertebrate, like mouse, for example, you can also use the orthologous genes from human for the analysis. So this would be selecting this option, this would then also allow you to use the annotation types that are available for human, like here the canonical signal transduction pathways. I just switch back here to human. You get a p-value for the overrepresentation of the different terms and different categories. You can change the threshold here, by default 0.01. You can also let the system calculate an adjusted p-value with a Monte Carlo simulation. The default for that is 0.05. I won't do it for this example, as it will actually not change the result in this case, and just will take more time to run. And you can also optionally upload a user-defined gene universe. So for example, if you use arrays with a limited set of probes and you want to include only those genes as the background set that are actually addressable by your array. In this case, as we have data from an RNA sequencing experiment, that just include everything we have in our system. Okay, I just give, I provide a name here for the result, and then I can start it. By the way, you can also let the system run the analysis in the background, which is, well, in this case not necessary, because it should yield the result within, well, maybe 1 or 2 minutes. While this is running, one thing before that we actually get that is we have one page here following the start, as the system is not sure about every search term that we have in our input file here. So these two identifiers are not clear to the system, so it makes an educated guess what you actually could mean with it, which is okay here in these two cases. And every identifier that cannot be found in the database is just listed here and will be ignored in the following analysis. Okay, so we can actually start the gene ranker analysis. And sorry, while this is running, I will show you a few more slides, just 2 more slides on the data set that we use here. This is a public data set from Gene Expression Omnibus. You see that's the GSE number is 87312, came out last September, and this is both RNA expression data from an RNA-Seq dataset and microRNA, non-coding RNA data. And we have the data actually come from a papillothyroid cancer cell line, TPC1 cell line, which stably overexpresses the sphingosine kinase 1, SPHK1. And this SPHK1, the sphingosine kinase, has a well-established role in a number of different cancers. It is known to phosphorylate sphingosine to sphingosine 1 phosphate, and the sphingosine 1 phosphate is then bound by a number of different sphingosine 1 phosphate receptors. These are S1PR125, which you see here. These receptors are G protein coupled receptors, and they're activated by binding the sphingosine 1 phosphate, and these activated receptors can then bind a number of different gene proteins, which you see down here, and activate, for example, cell migration, cell proliferation. And it is known that sphingosine 1 phosphate induces migration in carcinoma cells. So that's for the background, and we will have a thyroid cancer cell line which actually overexpresses SPHK1 as it has been introduced in an expression vector. Okay, let's switch back to our result, which we have here. Okay, so what does the pathway system result page and user interface consist of, on the one hand, we have control bars. You see one control bar up here, and you have another control bar down here. The upper control bar can be used, for example, to change colors in the networks. It can be used to find elements in the network, so you can search for a specific gene. Well, this is empty now because we don't have any network yet. And you can also export and import data into the system. So for example, you can export the list of genes that you have in the network here, or the connections you have in the network. You can export the networks in a number of different formats also as a graph. And the control bar down here is for the manipulation of the network. So you have a number of different options here for extending the network. You can, well, remove nodes here, specific nodes. You can change the layout, and you can also change the basic settings here. The most important thing to start with is the gene ranker list, as it is called. So this is actually the list of the different categories with the overrepresented terms in each category. You see the first one here is open, the canonical signal transduction pathways. Then we have literature-based pathways. We have the different gene ontologies, so you can just click on one and then you see the corresponding terms. And the terms are sorted by the p-value, so the smallest p-value is here at the top. You see the number of genes that are annotated with the corresponding term. For example, here beta-1 integrin sulfose interaction pathway. The number of genes that are in total in that category. So input gene 16, total number 66 in this pathway. Then you have a small info button here that if you click it, shows you the gene symbols and the gene names in that category. You can copy them to the clipboard if you want to use them in other programs. Okay, you can also get the complete list of the gene ranker result by using the element that you have down here in the lower right-hand corner. You can open this by clicking on this arrowhead and get the gene ranker results. I'll show you what it looks like. Here you have an overview again of all the different categories. Just a little different layout here with the list of the observed genes and the gene IDs. You can download the whole input gene list, and you can download also the complete result in an Excel file or in a text file in a tab separated format. And you can also access the complete list of genes which we have in each of the terms by just clicking on this link, which will then open a file. Let me just get this in here. Which just looks like this, a list of the identifiers of the genes in the category. Okay, so that's just for the overview of the data that you have in here and which you can download. Okay, so in the signal transduction pathways, we have actually two different ways how we can display the gene. So you can just click on such a term, or then you will get the genes as co-citation, as a co-citation network based on the co-citations of the genes in PubMed abstract. So the genes need to be co-cited together in one sentence in a PubMed abstract. Alternatively, you have this P button, which shows you the complete canonical pathway, including also those genes that are not in your input set. Canonical pathways come from BioCarta, from NCI Nature, from Reactome, and from INOH. And yes, we will shortly actually look at one of those. Before we do that, I'll just show you one other thing, namely how you can filter this list and how you can access a hierarchy of gene ontology terms if you go to the gene ontology categories here. This is another button that you only have within gene ontology. To filter this list, you can enter text down here. By default, the list is filtered by the name of the annotation that you have here by the term. And for example, we can use, if you change to the sequence transduction pathways here, we can just enter metallo, which automatically filters the list for in this case we have two entries in the pathways here, literature mining pathways. We have another two here under molecular functions, and we have another three here under pharmacological substances. Alternatively, if we remove that, get automatically the full list back. Alternatively, you can also filter for genes. So you get only those terms where a specific gene whose symbol you enter down here is included. You do this by toggling here between the search for annotations and genes. So this changes to gene. And if I enter the sphingosine kinase 1, which is the overexpressed transgene here in this setup, I also find a number of terms that include these genes. This gene, you have the numbers here, so this just tells you 0 of 3 available terms are selected at the moment, or 0 of 12 as you have them here in the next category. Okay, now if you look at the biological processes here, after we filtered for the sphingosine kinase 1, we see, for example, here in cell migration, which is one of the top terms in here, and includes 77 genes. It's not overly generic term. You see there are other terms here at the top that are more generic, like organism development or system development, etc. You can show the position of this term in the gene ontology tree by clicking on this symbol here, show the hierarchy. And this will then point at the corresponding term here in boldface, cell migration. You can also open the corresponding hierarchy. What's actually below that? Green means it's an overrepresented category. The deeper the green color is, the smaller the p-value is. You see here with larger p-values, it just is a very light green. And you can also open a network directly from here. So for example, regulation of cell migration, which has a p-value of approximately 10 to the minus 12. You just double click on it, and this will then generate a network for you. details about the networks with another network actually. So let's start with, just remove the filter here. Let's start with the canonical pathways. So we're now in the second part, networks. Canonical pathways, how do we display them? I mentioned that you can use this P button for it. I'll open the second one here, validated transcriptional targets of AP1 family members, FRA1 and FRA2. These two genes play a role also in cancer. FRA1 is a known driver of metastasis, and FRA2 increases the invasiveness of cancer cells. You see we have a number of nodes in here. Some of them are colored with an orange or a red background, and the other ones have a gray or maybe a white background. I just zoom in here using my mouse wheel. The default layout for the network is the so-called cell layout or cellular layout, which shows you the nodes, the genes in the corresponding cellular compartments here in the nucleus, the cytosol, the membrane, and in the extracellular space. This generally takes relatively a lot of space, so you can also change the layout, for example, to the hierarchical layout, which tries to arrange the nodes in a way so that you have the upstream elements of the network or of the pathway at the top and the downstream elements at the bottom. This also shows you protein complexes, genes, gene nodes which are shaded or are connected by a gray shade here are part of a protein complex. That's also the reason why you can have one and the same gene several times in this network, because it can change the protein complexes. This is not the case if you use the alternative view, which shows you the co-citation based networks. You can access information about interaction between genes and also information about the genes themselves. I'd like to pick out one example here, the connection of this complex FOSL1 and Jun to MMP9. FOSL1 and Jun both are transcription factors, and we have a binding site definition for these transcription factors in our system. You see it here in the tooltip under matrix families. It's called V$AP1F. Actually, for both of the two factors, so the two factors interact with one another and bind the AP1F binding site. Jun can also bind another binding site here, which is called V$CRAB. And the connection here from this complex to MMP9, which is shown as a target here of this complex, as can be, if you do a double click on it, you can display a bit more detailed information about this connection. You get information about the literature that is behind the connection. We have, for example, expert annotation for this connection that FOSL1 regulates MMP9, that it actually activates MMP9. You have automatically extracted information from the literature. This is called, these are the citations on so-called function word level, which means the two genes are co-cited within one sentence in a PubMed abstract, and there is also a function word like inhibits or activates or regulates between the two genes. We have 19 co-citations here for this connection on function word level. We have 36 co-citations on sentence level. This is similar to this, only the function word is not mandatory. We also have information that We have from mapped inspector, from the automatic annotation of the promoters of the target gene, that we have actually FOS-L binding sites in the promoters of MM9. If you want to have information on a gene, you can double-click on the gene node itself. You get some functional information here from public data sources. You can directly jump to the genome browser in the system. I will not show that today. And you can, for example, get information about the promoters under DNA sequence-based information. There's a lot of other information. I cannot show you everything today, but just to know that you can get detailed information about each gene in the network. Example, promoter regions. If you click here, this will open a new page. We have a list of the promoters here with promoter IDs within our database. The first promoter here is probably the main promoter of MMP9. It controls a large number of transcripts. And we can do a quick analysis of the promoter just by clicking on the link that we have here. And this gives us a standard MAT inspector search for transcription factor binding sites. And as you remember, we have an AP1F binding site in the MMP9 promoter, or at least one actually. You get a list of the matches for the transcription factor binding sites here, and you can also filter specifically for AP1F binding sites. So we get 4 matches in this promoter for the corresponding binding site. The matches are found both on the plus and minus strand, and the first two matches here represent actually one binding site. You see the strand information here. This is a minus strand match, a plus strand match, and the next two also represent one binding site where we have a match on both strands at the same position. You can also visualize that quickly here in the graphical view. This gives you a graphical view of all the transcription factor binding site matches, in silico binding site matches within this promoter. If you want to see only these AP1F binding sites, then you can deselect the binding sites here and just select from the list that we have on the right-hand side, AP1F, and you quickly see that we have a binding site here with a match on both strands and another one here also with a match on both strands with some additional information. I won't go in too much into the details here, just to show you that you can quickly get from the network to the transcription factor binding site analysis of the corresponding promoters. Okay, so the next thing that I'd like to show you is some information about the network connections actually. And this will be shown at the hand of another example, namely now not a canonical pathway, but a literature-based pathway. And I will use this TGF-beta pathway. You see there is no P button down here, but you can just click somewhere here in this on this entry, and this will now generate a network based on the co-citations. We see that we have both upregulated and downregulated genes. So upregulated again in red or orange. You also see the measurement values here, the log fold changes in the tooltip. So 1.925 here for this LDBP1, or this one with a darker red or stronger red, 4.415. Downregulated genes are in a blue or purplish color, like one here minus 2.6 and this one here minus 4.5. You also see that you have a number of different types of connections here between the genes. So these connections are based on the co-citations by default. On those, you get those connections with the strongest support, with the highest number of co-citations, with the highest number of co-citations that are also expert curated. And you have, you see that you have different arrowheads, you have different lines. You might have solid lines like the one here, you may have a dashed line like here or a dotted line like here. And all these different things mean something. A solid line means we have actually expert curated annotation for this connection. A dashed line means we have information from the ENCODE data and also from our own chip sequencing data that we collected from the literature, that there is actually binding of a transcription factor to the target gene here. So for example, if you look at this one here, BLHLHE40, a transcription factor, which is shown here also by the sequence logo just below the note that it is a transcription factor for which we know a binding site. We have a validated interaction with a regulatory region of this gene cited 2 from ENCODE, very probably. You can actually check where it comes from. If you do a double click here and you have click on this validated regulatory interaction, you see that the source for this is from ENCODE transcription factors, and it also shows you in which cell types we have binding of the factor in regulatory region of cited 2. Another thing here, if we look at the connection between these two genes, TIMP1 and ADAM12, we have information that this is actually an inhibitory relationship shown by this specific arrowhead here with a circle and a bar. You can also double click again here and get this information. TIMP directly inhibits activity of ADAM12, and you can also access the PubMed abstract. If you click on the link here, then the corresponding abstract will be loaded so that you have direct access to the underlying information. Okay, as an overview for all the different types of arrowheads and arrows, etc., you can click on this GePS legend, and this will show you a number of things. Also here the shapes of the different nodes, the color, some other information, what these numbers mean, and also information again here for the arrows. And one more thing here, if an arrowhead is filled with a black or a gray color, this means that we have either a validated binding site in black as we see it here, or at least an in silico binding site in gray as we see it, for example, here. So this would mean in the BMP2 promoter there is an in silico binding site for the EMX2 transcription factor. Okay, one more thing about the transcription factors. In the network is a tabular overview of the transcription factors, which you can access again using this element down here. The first option here, gene TF analysis for the network genes. So these are all the genes that are presently shown in the network. will give you a table where you have a column for each transcription factor. Just need to click here. A column for each transcription factor in the current network, and a line for each gene in the current network, including the transcription factors. And this gives you an overview of the information we have available for the interaction of these transcription factors to all of the genes. For example, here for the BH-LHE40, so this guy here, we can select, we can sort the list by clicking on the arrowheads here, and we find that we have 7 entries with a dark blue background, which means that we have both a validated interaction from ENCODE data, for example, symbolized here by the first plus sign that we have. We have an in silico binding site match here symbolized by the second plus sign, and we have a co-citation symbolized by the third plus sign that we have here. And it also tells you for the co-citations on how many co-citations we have on each level. So here we have 1 co-citation on sentence level, or for this interaction here with GDF15 we have 6 co-citations on sentence level, we have 2 co-citations on function word level, and we have 1 co-citation on expert level, and we can also access this information by just clicking on the link, and then this will be shown in another program from our Genomatix suite, the Lit Inspector, again with links to the original abstract. Here, the green ones here, they don't have a validated interaction, so nothing from ENCODE, but they do have an interaction or have a in silico match, and they have also a co-citation, and the rest here does not have any co-citations with well varying amounts of the other types of evidence. You can see an overview of what actually is what down here. Okay, so much for the overview for the GNTF analysis. And now we come to the last chapter, namely how you can manipulate networks. We will start with this one here, matrix metalloproteinase network. So the first thing that will change is the basic, some of the basic network parameters. You can access these network parameters down here in the settings dialog, which you open with this settings button, the one with the cogwheel. There are a number of different parameters that you can set. I will not explain everything. If you use the system, then just you can have a look at the help pages. I'll just show you 3 things here. One is the minimum evidence level. I mentioned that by default we have the sentence level for the co-citation of 2 genes in the network, and we can change the stringency here. For example, if we go to the function word level. So this is a bit higher stringency. We can change the network type from simple network to shortest path network. The difference is which of the connections are shown preferentially. Not all of the connections between all of the genes can be shown in the network, but it would just, you would be overwhelmed by all the connections. So only the most important ones are shown. In a simple network, the strongest connections are shown. In a shortest path network, and not necessarily the strongest connections but are shown, but the connections from the strongest hubs, so to speak. So from those genes that have connections to the highest number of other genes. And the shortest path network uses one gene as a seed. If you don't select the gene yourselves, then the system chooses it for you, and it takes the node which has the highest number of connections to different other genes, and the connections are then drawn in a way that the shortest path, or the shortest possible connection from each gene in the network to this seed gene is displayed. The system also generates hidden connections which are only displayed if you click on a node, and the number of these additional hidden connections that you can show by clicking on a node is defined by the parameter here. The default is 3, and we can just also change this so that we get up to 10 additional hidden connections that we can show optionally. And then we can save these the settings, and in order to apply them to the current network, we just click on this network generation button down here. It also says here it generates now a shortest path network and not a simple network. And it automatically finds MMP9 as a hub where the network actually gets started and shows the shortest connections to all the other genes. Now, as we also have microRNAs in this data set, we might want to include some of them. Even if they are not annotated with matrix metalloproteinase one way or the other. And for this we can add, or we can select down here from these buttons which allow us to extend the network with different types of nodes. The third option here allows us to select microRNAs, or actually to let the system select microRNAs. So if we click here, I can select the number of microRNAs which should be added, and also with how many of my input genes the microRNAs need to be connected in order to be included here. I changed the number here from 10 to 20. And it's okay, it's enough if the microRNA is co-cited with only one gene. And this now searches for one in my input set. I also have microRNAs in my input set, but it also searches in the literature. So preferentially it will add genes from my input set, and if there are not enough there to count up to 20 here in this case, the literature is searched. We get the information about the microRNAs here, and we don't see which of them are actually from our input set and which are just from the literature, because there is no coloring of the nodes, because, and this is the case because none of them are in this category here. But we can apply a small trick in order to show the color for the microRNAs as well. Namely, there's another category down here that's called more gene lists, and this just lists all of your genes, or just the upregulated or downregulated genes. Now, if we just activate input gene list here, as for all of our input genes, and change the logical operator, how these filter terms, well, metalloproteinase pathway here and input gene list here, to OR instead of AND, we now get colors for all the genes, the microRNAs which are in our data set. Okay, so in this way we can add microRNAs, and you can, for example, look at the microRNA 144, we can actually search it in here. Mir144 is here, so this is for finding a gene, and you can, for example, you see, for example, that this is connected to PTGS2, and we have information here from the literature that PTGS2, which is shown here as cyclooxygenase 2, which is a synonym, is actually a target of microRNA 144. So one more thing, adding genes manually. In order to just manually add any gene, you double click somewhere here in the white region and you can enter a gene name or a gene symbol. I'll enter the sphingosine kinase 1 once more. The system searches in the database whether it finds anything. It might take a few seconds until it's found it. Here it is, and then you can just select from the list here and you can click OK. And the kinase will be added, as it is also in our regulated list. It's upregulated because it's the transgene in the experimental data set. We get a red color, and we can include it in the network here. But before we do that, let me just show you another thing here, namely removal of nodes. You can remove nodes based on, well, their specifics. For example, removal of filter genes, which is the first option here of these three, will remove all these microRNAs which don't have a color. The second option will remove anything that doesn't have any interaction. So if I use that, this would be removed because we don't see any interaction yet. Let's just remove the microRNAs that are not part of our input set. This is always connected to a recalculation of the network. Okay, now we get only our input data. And if I now want to see the sphingosine kinase as the center of my network, to see the connections from sphingosine kinase to all the other ones, I just select it and again click on network generation. Now my sphingosine kinase is the seed of the shortest path network. And the connections will be shown accordingly. In order to better see the different hubs that we have here, so sub-hubs, we can now change the layout to the centric form. And now we have the sphingosine kinase here in the center. You see here in the overview also nicely the central hub here and some sub-hubs here, 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 and here, which is then also see here and here. So for example, we find that the PTGS2 again, so the cyclooxygenase, is a target of sphingosine kinase, shown here with this activating arrowhead. And also if we double click here on the connection, this is also shown here as PHK1 indirectly increases the expression of PTGS2. And this microRNA 144, as we already saw it, is also connected to PTGS2. So PTGS2 is a target of this microRNA. It is known from the literature that microRNA 144 together with microRNA 26A inhibits metastasis and tumor proliferation. So it could be the case, because this is downregulated, that it contributes to metastasis and proliferation, maybe by now de-repressing PTGS2. Okay, so we don't have any of this information directly visible here in the network, but we can add it if you just click and drag from the microRNA to the PTGS2. You can create your own interaction. By default, you have an automatic determination of the interaction type, but if you want to add it manually, you can just select from the list here, microRNA 144 inhibits PTGS2. And then this will be shown as an orange connection with a corresponding arrowhead here. Similarly, if you want to add an information, an activating information here between SPHK1 and MMP1, we have the information, if we double click here, that SPHK1 regulates MMP1 and actually activates it. And so we do this again here, and we tell the system that SPHK1 activates MMP1. Now this can be saved only in this network actually, so not in a database, but you can save this view including your additional arrows by choosing this button up here, save, load and delete a GePS session. You can give it a name like my network, save, and this will then be available with all the settings that you currently have later on in another session. Okay, the last two things are network extension with transcription factors and adding connecting G nodes to a network. I'll show you this at the hand of another network, namely the mothers against DPP homolog, so SMAT pathway actually. So this means we have genes that are co-cited with genes that are in this SMAT pathway. Among the SMATs, we actually find only SMAT6, which is one of the inhibitory SMATs. The other ones are not in the gene list of upregulated or downregulated genes. Sorry, but we can add SMATs to this network. We do this again using these the add elements box here, but now I just add a list in plain text. So I add SMAT1, SMAT2, SMAT4, SMAT5 and SMAT9. Add to list. Now they are added like this, and I can incorporate them into the network by just clicking on network generation. And if I change the layout to hierarchical, it's a bit nicer. And I'll see my SMATs here in the network. I see among other things that the microRNA 146A is a regulator of SMAT4 and SMAT2. I find that the BMP2, which is a known upstream regulator of the SMATs, is here connected to SMAT9 and SMAT5. I see an activating relationship here to SMAT5. I also see a activating relationship to SMAT4 if I zoom in here a bit. So this would be this connection, which tells us that BMP regulates SMAT1 and actually increases the activity of SMAT4, also of SMAT4 here. And similarly, if we look at the BMP6, we also see a connection here to SMART5, for example, and also here to SMART1. Okay, now the last thing, how can you connect genes via shortest co-citation path? I'm going to add automatically a number of transcription factors to this gene here, SLPI, which is one of the, well, strongly upregulated genes. Again, I use one of the options down here. Here, extend by transcription factors, so not a manual addition but an automatic addition. I only take 10 here, extend the whole thing, and then try to find, well, a possible way from my sphingosine kinase to the SLPI1. I have 10 added transcription factors here. I'll just pick out the NFE2L2. As I already know that I get a connection here. I add my sphingosine 1 kinase once more. And then I let the system, as soon as I get it, I let the system search, here it is, for a connection, the shortest path connection of SPHK1 to this NFE2L2. And for this, I just select the two genes and choose this option down here, connect the genes by shortest paths of co-cited genes. Now the system will look into the database whether the two genes are directly connected or whether they can be connected with one jump using another gene, and this is the case for the PTGS2 actually. So we have a connection from SPHK2 to PTGS2. We see that PTGS2 is, I can add the color once more here, is a target of SPHK1. Here we also see something in the other direction. So PTGS2 is also a target of NFE2L2, but if we look at the function world level co-citations, we find one or two. One of them is this here that also gives us some information that NFE2L2 can be regulated by PTGS2. So we have a potential way from here to here to here. And then if we look at this information where we have a confirmed binding of the NFE2L2 transcription factor in the promoter of SLPI1, this could be another connection. Okay, so much for a rather dense overview of the Genomatix Pathway System. That is, well, actually we are already over time, so I just check here for questions. Yes, there was one question here. What does 77 mean in 77 of 1,777 genes? This is from here. So we have, this means that we have 77 genes in our network, in our uploaded list, which are annotated for the specific term here. And of 1,770 would mean that we have in total 1,770 genes in here. This could be the case, for example, if we have a very generic term. Okay, I don't see any questions anymore, so I'd like to thank you for your attention. If you'd like to test our system, you can just go to our website www.genomatix.de, and you can click on here on this free trial button, and this will then you will just be asked to enter some information about yourself. You will get a 1 week free access to everything with no restrictions, and then you will be asked to answer a few questions on the questionnaire, and then you can get another week of a free trial. And there is a recording of this webinar, so everybody who registered will get the recorded webinar within the next 2 days at last. Another question here that I get, do you have a feature of checking how does the system react to mutation in some gene? No, unfortunately not in the Genomatix Pathway System. Okay, so thank you very much once more, and well, maybe see you or hear you next time when we have another webinar. Bye bye and have a nice day.